So welcome back. Today we talk about intraembryonic mesoderm and neural tube derivatives, and I am Dr. Gaurav Agnihotri. So we have done the formation of notochord. Notochord was formed by day 17 of intrauterine life. Now this is the mesoderm on either side of notochord shown by these blue dots. Now what happens is mesoderm on either side of notochord thickens into paraxial mesoderm. So mesoderm in this region has thickened to form this paraxial mesoderm. More laterally is the lateral plate mesoderm. This one is the lateral plate mesoderm, and in between is intermediate mesoderm. So students, what happens is that these blue dots which represent the mesoderm, the thickening takes place, and we have now got from medial to lateral side paraxial mesoderm, intermediate mesoderm and lateral plate mesoderm. Now paraxial mesoderm undergoes segmentation to form somites, which are structures triangular in cross section. Each somite undergoes division into three parts: dermatome, myotome and sclerotome. Dermatome forms dermis of skin, myotome forms skeletal muscle and sclerotome forms vertebral column and ribs. So now we can appreciate that from which part of mesoderm the skeletal muscle is formed. It is formed from paraxial mesoderm. So paraxial mesoderm, in this area, it undergoes segmentation to form structures which are triangular in cross section called somites. Now each somite undergoes division into three parts: dermatome, myotome and sclerotome. Dermatome forms dermis of skin, myotome forms skeletal muscle, while sclerotome forms vertebral column and ribs. So these are the derivatives of the somite. Dermatome forms dermis, myotome forms skeletal muscle and sclerotome forms vertebral column and ribs. And from where are the somites derived? They are derived from paraxial mesoderm. The intermediate mesoderm, this one, it forms urogenital structures, the kidneys and the gonads. Lateral plate mesoderm, it develops a cavity. So small cavities appear which unite to form a large cavity, and that is called intraembryonic coelom. With the appearance of intraembryonic coelom, this lateral plate mesoderm now gets divided into splanchnopluric intraembryonic mesoderm towards the endoderm and somatopleuric intraembryonic mesoderm towards the ectoderm. Now we come to splanchnopluric intraembryonic mesoderm. So this one here, which is in contact with endoderm, it forms the heart and blood vessels, and we state that the heart is formed in splanchnopluric mesoderm forming floor of intraembryonic coelom. So in this area the heart is formed. Now somatopleuric intraembryonic mesoderm, it forms lining of the cavities, the pericardial, pleural and peritoneal cavities. So somatopleuric intraembryonic mesoderm forms the lining of the cavities, and the intraembryonic coelom by itself, it forms the pericardial, peritoneal and pleural cavities. Now the unsplit part of intraembryonic mesoderm, this unsplit part of the lateral plate mesoderm, it forms the septum transversum. Its cranial part helps to form the diaphragm, while liver develops in its caudal part. So diaphragm and liver, they develop in relation to the septum transversum, the unsplit part of the lateral plate mesoderm. The neural tube now, how it is formed. This notochord, it acts as an inducer. It stimulates the overlying ectoderm to get thickened into a neural plate, and then this neural plate, it forms a groove which is known as neural groove, and then this groove pinches off to form the neural tube. The cells at the junction of the groove with the rest of the ectoderm, they form the primordia of the neural crest, shown by cross here. So this is the junction of the neural groove with the rest of the ectoderm. So with separation of neural tube from surface ectoderm, cells of neural crest appear as group of cells lying on dorsolateral aspect of neural tube. We call these the primordia of neural crest. Now this is the neural tube formed. Its formation has been induced by the notochord, and the primordia of neural crest are lying on the dorsolateral aspect of the neural tube. Now this neural tube undergoes subdivision into an enlarged cranial part, which is destined to form the brain, and a narrow caudal part which is destined to form the spinal cord. So this cranial part, it gets differentiated into prosencephalon, mesencephalon and rhombencephalon. The prosencephalon further gets differentiated into telencephalon and diencephalon. Telencephalon forms the cerebral hemispheres, diencephalon is the thalamus and hypothalamus. The mesencephalon forms the midbrain, while the rhombencephalon gets divided into metencephalon and myelencephalon. Myelencephalon forms medulla oblongata, while metencephalon forms pons and cerebellum. The process of formation of neural tube is called neurulation. Now coming to the derivatives of neural crest. So sensory ganglia, spinal ganglia and autonomic ganglia, they are all derived from neural crest. Sensory ganglia of cranial nerves 5th, 7th, 9th and 10th. So sensory, spinal and autonomic ganglia, and then other components of the nervous system, Schwann cells of peripheral nerves, pia mater and arachnoid mater of brain and spinal cord. So these 5 structures of the nervous system, they are derived from neural crest. Most structures are derived. These are the cells, odontoblasts and the melanoblasts of skin, and odontoblasts, they are also derived from. So odontoblasts and melanoblasts of skin, they are derived from neural crest. C cells of thyroid and chromaffin cells of suprarenal medulla, they are also derived from neural crest. So these 4 specialized cells, they are also arising from the neural crest. Then we have got connective tissue and bones of skull and face, and dermis of face and neck. So these are derived from neural crest, and smooth muscle cells in blood vessels of the face, they are also derived from neural crest. So remember these 12 structures which are derived from neural crest. So that brings us to the end of the topic, and I want to end by stating that by the end of 3rd week, neural groove is formed. By the end of 4th week, neural tube is fully closed. So till the next time we meet, it's bye from my side. So keep following the lectures and do give me a feedback. Thank you.